scientists there to teach me stuff and teaching me how to do it hands-on, teaching me processes. And it's quite interesting because I've been to labs like quite a lot in different places around the world, and so I see very interesting differences and things like that as well, which is lovely. So at the moment I'm here working with the Seagull lab at the Weizmann Institute. I said it, yeah, I've been practicing. I nearly forgot. So yeah, I'm working with the Seagull lab there and looking at things to do with my gut microbiome. And this week I shall be sequencing my gut microbiome and then developing art around it, which you'll see connects to quite a lot of other work I've done. So this is a small detail of an earlier work, which is part of my original normal flora project. So the term normal flora, it's kind of it's not a very popular term anymore, but it sort of means the ubiquitous bacteria and molds that we coexist with. It's kind of been changed now to, it's kind of been rebranded, I guess, as the microbiome. And it's been interesting to kind of observe that change over time. So originally as an artist I was interested in the limits of knowledge, I guess from a kind of philosophy of science perspective, like where do we draw the line with what we study? And I was told that normal flora bacteria weren't studied because they were of no commercial or medical interest at the time. Now this might seem ridiculous to you now because the concept of the microbiome is almost like a gold rush in science now, where we have these new tools, these sequencing technologies, relatively new tools, in order to help us look at these things. But in those days, it wasn't just like looking for a needle in a haystack, it was like looking for loads of needles in millions of haystacks, to use an English metaphor there. But it was there was a lot of stuff out there, but you would never find it. So there was a lot of bacteria that they just weren't studied. So I decided I would map the bacteria in my domestic setting. Now this sounds pretty normal now, but in those days people didn't do it at all. And I went and started to work with a microbiologist at Brighton Hospital, who was the consultant microbiologist. But now he's the regional microbiologist for Public Health England in the UK, so he's in charge of the whole southeast of England for heading up microbiology services for clinical microbiology. And when I was introduced to him, another scientist who introduced me to him said, "Anna, don't do that thing you do. Don't go into the lab and tell him all your crazy ideas, because he's quite normal and you'll scare him." And it turned out I sort of digged anyway, and he wasn't scared. In fact, he loved it, and he's not normal at all. And he's been all around the world with me giving lectures on, he's started to get into research on similar thing as me, which is around this notion of in aesthetics theory, the idea of the sublime in relation to bacteria. And he's given lectures on this at all sorts of like media art conferences and things, which has been amazing. But for this piece, so I started to swab the bacteria in various corners of my domestic setting, my house, and I would plate them up on Petri dishes, do their subculturing, and then use these tests, biochemical tests called API tests, to kind of look through everything. And then when you've got this kind of numerical code, depending on whether different pockets in the biochemical test change different colours, you type this into a computer, and it would say 65% chance Staphylococcus epidermidis, or something like that. Can you imagine how imprecise this was? And then when you were looking for bacteria that weren't, commonly known, you'd get like 20% it's this or something, and you'd be like, there's no chance you're going to find out what this is. But nowadays we can find that out. So that was really interesting. And once I swabbed something in the corner of my bathroom near my shower, and we looked it up. I can't remember the name of the bacterium, but it said, when we looked it up, it said an emerging pathogen in catheter infections. And I said to the scientist, what's it doing in the corner of my bathroom? And he said, well, I suppose nobody bothered to look for it before, because you wouldn't. So that was where I kind of got my grounding in this work. And this piece is The chair is carved with the images of the bacteria that were cultured from it, and the needlepoint seat embroidery there is the light microscopy of the bacteria cultured from it. And then this crochet here is a kind of quite large now, growing crocheted piece based on the Staphylococcus epidermidis that I cultured from my bed, based on scanning electron microscope images. And I do this as a participatory work, so other people, when they come to an exhibition, they join in with it. So there's balls of wool next to it, and you can add to the crochet whilst chatting about your ideas about bacteria. You know, like your questions, you can talk to me about that and things like that, and we have images available and things. And what I found is that really bad crocheting is really lends itself to how Staphylococcus epidermidis looks. So that's quite lucky. It's quite big now, and when you so when I show it, every time I have a solo show, I include the piece, and there are balls of wool there, and I feed it with wool, and I feed it with gallery visitors, so it kind of grows exponentially to fill the space of the gallery, depending on the number of visitors, which I kind of consider the sort of the things that metabolize the food, and the wool is the food. So it kind of, if you think of it, it's like a metaphor for bacteria as it grows. So that was quite an early piece, and there's a whole series of things about that project. And then I'll come on to this one, which was, it's called Infective Textiles, and it was the first piece where I created a huge kind of petri dish and was growing this dress. I won't go into the history of why it's a Regency ball gown and things, but there's a whole story around it, and to do with the sites which I swabbed for bacteria. But we made a kind of 4-meter petri dish out of plastic sheeting and garden cane, plasticine, and we made the agar-agar from a do-it-yourself recipe that Simon Park, who's a microbiologist, created. And we basically bought up all the agar-agar in the local Chinese supermarket. It's quite a few years ago. And they're like, "What are you doing with this stuff? We cleaned them out. We needed vast quantities of it." And plated the textile on top of the agar-agar and cultured it with bacteria from various environmental sites that were related to the story of the artwork. The embroideries on it, I worked with a scientist called Melissa Grant at University of Birmingham, and the embroideries are, she works in the School of Dentistry, and she was working at the time on a toothpaste that was made from the active molecules of natural antimicrobial substances. And so she was a great resource for telling me, well, this one might be antimicrobial, this thing might be antimicrobial. And I discovered like a lot of dyes, like natural dyes are antimicrobial. Woad is, madder root is, safflower is antimicrobial. So I worked a lot with that. And so where we embroidered on the dress, patches of embroidery, the bacteria wouldn't grow, or sometimes, depending on whether it was antimicrobial to it. But that was quite an early one. It would be brighter colours if I was able to leave it in the agar for longer. It was made as part of a live open lab with a project called Laboratory Life, which was Lighthouse Gallery and the Arts Catalyst in London organised. And so it was time limited. The piece grew over about 8 days, and then the exhibition opened on the 10th day. So so it was quite, and I was made with a group of other people who worked with me on my project, and then we had to have it all open to the public. But the actual lab was also open to the public, so the whole time that it was growing, it was open to the public, so they could come in and see the bacteria growing, look at bacteria under the microscope and things like that. So that's interesting from a health and safety perspective, because people, we're having to kind of negotiate that for sort of the first time of showing something with such a large amount of bacteria just growing there. But luckily, working with the head of microbiology for Public Health England for the South East, where it was based, he could go over all the protocols and check that. So the health and safety was already taken care of. Very important to me. In fact, I've written a book on the ethical implications of artists working in labs. So this explains it a bit better, the technique. So this is my Mercer quilt. I call it MRSA because I'm English, but I understand it's pronounced Mercer here. I was corrected today.
It's both.
So the blue on the quilt squares, this is just a detail of it, it's a full-size quilt. The blue is the bacteria grown on chromogenic agar, and then the patterns, like the polka dots there, are different tools and techniques and treatment and diagnosis of the disease. So you've got, this is a cafoxetine strip here. This is vancomycin diluted as if it's an intravenous, if you were having an intravenous treatment with it. And these are vancomycin susceptibility discs that they use in the lab to see whether it can be treated with vancomycin. And up there, that's an interesting one. So the embroidery up there, that's turmeric, the spice. And you can see it has a limited, what they call zone of inhibition, a limited area where the bacteria hasn't grown up to the turmeric. But not brilliant, so it's not going to solve anything yet. I'm obsessed with the history of tuberculosis, and I've done a huge body of work around that, which is called the romantic disease, which might sound quite a strange name depending on your perspective on the history of it. But in the UK, it was considered really quite fashionable to have tuberculosis. Does this seem very weird?
Yes.
Have you ever heard of the romantic poets, Lord Byron and Keats and people? Well, and have you ever come across the term pale and interesting to describe somebody? Have you ever heard of that? So it's sort of, oh, she's very pale and interesting. It's like, means less sensitive, stay indoors. And Byron said, he declared, "Oh, how pale I look. Do you know, I think I should like to die of a consumption," which was the old name for tuberculosis. And then all the ladies would say, "Oh, how interesting that poor Byron looks in death." And it was kind of, they almost wanted to have it. So it's almost like, I'm from Brighton in the UK, and you sometimes have a similar thing with HIV, where people kind of want to have that badge. And with the romantic poets, it's sort of very tied up. So I've read, I collect a lot of historical things, and I've back from Galen, the most ancient kind of medic, he talks about something called the spes physica, which is a kind of state of euphoria in late stages of TB, where the patients are kind of gasping for breath, but you have these very kind of creative outpourings. And he also said that the patient, it means the hope of the consumptive, and the patients couldn't be convinced that they were dying, and they had a huge energy as well, so they wanted to kind of create. So there's this weird association with romantic poets, right up until like, well, even now to an extent. But right up into the 50s, I've got a document from an American magazine called The Voice of the Sanatorium, which is the hospitals they used to put TB patients into, and to keep them out of society. They believed in things like fresh air and things like that. And it says, there's an article that says, don't worry, you might have TB, but you might suddenly become a really good poet, because Keats apparently didn't write anything good until he got TB. And if you look into it, there's some quite interesting, even scientific studies. There's something called the genius germ hypothesis, you can look it up. And that correlates outbreaks of TB and leprosy, Hansen's disease, with creative periods, like paradigm shifts in Western sort of knowledge, I guess, which is kind of a very bizarre thing. So I'm not saying that it is. I'm not saying this thing is true, but I find it as an artist very intriguing that this kind of myth or kind of way of thinking about TB has grown up. And with a lot of diseases, but TB's one that's been with us from it is the oldest disease known to man. It's been found in Pliocene era mammoth bones, and it's still with us. It currently infects around a third of the world's population, of which 10% will get sick and 5% would die without treatment. And it's been a challenge for us throughout history really. So I find it fascinating, these myths that we've kind of created and woven around it, even around the treatment and the care of it. And I like kind of unpicking these things and how knowledge changes as well. So this piece is called Where There's Dust, There's Danger, which was the advice of the British Society for Prevention of Consumption in 1902, where they said that all passages should be kept clear of dust. Where there's dust, there's danger, because they believed that you could catch TB from dust. You can't. That's good news. But you can catch it from somebody with active TB coughing on you. It's like 99.999% of TB. So this piece is made with needle felt, but also incorporating dust. So it's the little pairs of lungs are basically made with dust. And it's impregnated with the extracted DNA of TB, because I'm fascinated in like objects that are kind of touched by disease. The patterns on it are the bronchia and stuff, but it's also got granulomas forming, and some of the lungs have had pneumothorax performed on them, which was a treatment for tuberculosis used up until the 1960s, which is where they would introduce air into the lung cavity to collapse the lung of the patient in order to give it a rest. Because rest was kind of the only thing that you had. So this piece is called Rest, 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 and this is based on a model that Birmingham Hospital in the UK, which was the TB sanatorium, big TB sanatorium, had in their had a photograph of in their archive. They did like patient or like public health days, and they showed these kind of little hospital beds where you would be in the ward. And I work with a lot of kind of antiques which I alter and change, and so this is an original toy hospital bed, and the screen. So the screen is dyed with madder root, which was used as an ancient treatment for TB. It's the oldest dye known to man. You'll see it in museums all the time, sort of pinky red. And the bed's stained with walnut husks, the nut, the outer part of the nut, which is also a dye which really smells like hospital disinfectant when you cook it, and it stains it brown. And then the pieces, again, it's tainted with the extracted DNA of tuberculosis, which I extracted in a category 3 TB lab and added to the artwork. So it's a in case you're worried, it's a fully validated protocol. I've worked with the director of microbiology for Public Health England on the piece, and I even did public workshops in Oxford Hospital in the UK, where visitors could actually add the extracted DNA of TB to little felted lungs made of dust and wool that they made themselves. This is a sculpture which uses an original pneumothorax machine, but I've carved it with the effect of lung tissue that's been affected with TB. And so you get, when it's got TB, sometimes you get these kind of dotty effects, and then you get these cavities appearing as well. And I went to study lung tissue in the dissection room at Brighton and Sussex Medical School in order to make it authentic. And the engraving on the cylinder there is based on they call it the characteristic scattered red ribbons that you would see. So that's what TB looks like under a slide. If you look at a light microscopy slide of it, it looks like red ribbons thrown down. So it's based on that. So the thing with TB, it's a really important challenge these days, especially for biology and people working in the field, because it's becoming extensively drug resistant in some places. So people are actually catching the drug resistant strains, because if you get infected by somebody who has actually got drug resistant TB, then you get drug resistant TB as well. And for some types, there are almost no treatments left. It's very hard to treat them. You can sequence the genome of it to see that it's extensively drug resistant, and you can kind of try to target the medicines, but it's incredibly difficult. And in a lot of places where they have a lot of extensively drug resistant TB, then the healthcare system is kind of struggling as well, so it's very problematic. So they've actually trialled pneumothorax again a few years ago in India, and it was shown to have a limited effect on extensively drug-resistant TB. So, I mean, we might go back to that if we don't kind of come up with new treatments for TB, which is kind of bleak. I was, on a positive note, I was working, well, I recently had a meeting with a new drug development team for tuberculosis at Dundee University in the UK, and they're working on it. There are people working on it. We'll see what comes out. Oh, I don't know. They pulled something out.
So you just isolated DNA from tuberculosis and you just put it on your a drug?
Yeah.
Okay.
Yeah.
A lot, a little?
Well, not that much because it's quite hard. You don't get a lot.
Right. Just kidding.
So I have some still.
Of course you do.
At home.
Right here.
Not TB DNA. I don't have any of that. Hopefully. I know I've been working with it, but I've been quite careful. So yeah, no, if you've got kind of questions, just ask. So this piece is actually called Don't Try This at Home, and it's at Eden Project in the UK, which has got a big exhibition called Invisible You, the Human Microbiome, but it's also got a huge biome as well. It's got the largest greenhouse in the world, which is a rainforest biome and a Mediterranean biome. It's very spectacular and well worth a visit. And they've just received quite a large amount of funding to turn another large building on the site into invisible worlds. So they're going to make an exhibition of all the kind of areas of science that are invisible or hard to talk about. So they're talking, because they're very interested in the environment, they're talking about things like water and food production and things, but they're very interested in things like synthetic biology and sequencing. But these are quite hard things to communicate to the public, so it's quite a challenge. Anyway, this piece is about human fecal transplants, which are quite popular these days, especially in America and places. But in the UK, it's I think they are done here, aren't they?
They are.
Yeah.
There's a bank. There's a bank in the hospital near near Lifeline Institute.
Yeah. So it's. We've got in the UK we've got something called NICE, and it used to be stand for the National Institute for Clinical Excellence. So it's something a bit longer now, but NICE are the people that basically decide whether the NHS can afford a treatment or not, and most of the time not. So it's a bit annoying. It's kind of a bad acronym, but under NICE regulations, you're allowed to perform human fecal transplants in the UK as a last resort for Clostridium difficile infections, when all other treatments have failed, when people have had at least 2 strong treatments of antibiotics, and that's failed. So C. diff is a kind of, so I was talking to scientists today at the Weizmann Institute, and it was interesting because they thought that the scientists in the UK sounded a bit weird, but you have to tell me what you think. So the people I worked with on this piece, they love Clostridium difficile. They think it's the most fascinating. What happened now? That wasn't me.
Maybe you clicked the...
I didn't touch it!
Maybe you?
That's not me. They love Clostridium difficile. It's an interesting microbe because it wasn't a human pathogen until 1972. It only evolved to be a human pathogen as a result of antibiotic use. So we actually caused it to be a human pathogen. It's just part of the normal gut flora before that. But then it took on drug resistance genes. And what happened was when you take antibiotics, certain kinds of antibiotics, that will kill off some of your gut microbiome, the bacteria that live in your gut. and kind of leave a space for this Clostridium difficile, which isn't killed by it, to kind of grow, I guess, like a weed. You know, like weeds in a garden, how they take over when you clear the space. So it's very much like that, and it causes terrible diarrhea and ulceration to the gut and things like that. So it's really problematic if you get it, because the only treatment.
Can you say maybe what it means to take on antibiotic resistance? So C. diff took on.
Yeah. So bacteria can evolve to compete with antibiotics that we treat them with, basically. So for instance, some of them will share these kind of bits of DNA that are like upgrades for them. So, I mean, I'm sort of dumbing this down a bit, but some of them have mobile elements of DNA which confer drug resistance that they can share between them. Others will evolve in different ways, and then we're sort of evolving our antibiotics to compete them. But antibiotic resistance has been with us since the beginning of time, because bacteria have just been growing and producing antibiotics to fight each other off since the beginning of time, so it's nothing new. I mean, it was well known even in the beginning. Fleming talked about it, and with streptomycin, which was the first really good treatment for TB. So the first cohort of patients, I think about 3 of them died of drug-resistant TB within a year. So it's the first time they got better, but then it came back. They didn't know how much quantity of drugs to treat them with and how long you needed to treat them with it. It came back, and then it was resistant.
By the way, the fecal transplantation for the Clostridium is more effective than the
Oh yeah. No, so the antibiotics are it's a vicious circle, because if you take more antibiotics to treat it, then you just get more drug-resistant bacteria. So you're like keeping it at bay quite a lot of the time. Yeah, but so the way it is at the moment is you're supposed to have like in the UK 2 treatments that have failed with antibiotics before you'll be considered for this, and you have to be really sick. The reason is that they don't understand the future effects of a human fecal transplant. So there's a lot of indications that it might influence other things. Weight gain is one. There's been a court case in the USA already.
It was approved in the US.
There is also the opposite. Gain weight loss.
Yeah.
Right? Maybe.
Well.
Probably not, but.
There's been a court case for somebody. Somebody said that they became obese after having a human fecal transplant from their daughter.
And they're both eating like the same food, so which is not good, probably, I guess.
Well, I don't know. I mean,
you can't blame it on the bacteria.
One of them wasn't obese before, so there's been a court case. So they're very wary of it anyway. I'm not sure if it's been approved. Maybe it's been approved recently.
It's been approved by the FDA.
Because we had people coming to the UK to have it, because it's quite. I think there's so much litigation there. Some, doctors are quite wary of it as well.
One of our friends did it to himself.
Yeah.
Josiah.
Yeah.
From the Odin, so you can read about it. This guy just did a full microbiome transplant for himself, in the mouth, in the ears, in the body, in the gut.
It's quite a strange protocol he did. I'd like to talk to him more about it, because it's
You can find him online. His name is Josiah, from the Odin, the Open Discovery Institute.
How did he select his donor? Yeah.
His friend. Yeah.
But he used antibiotics to kill off his original.
Yes, so that's how we did the transplant.
Seems really odd.
Yeah.
And even like his skin and stuff, doesn't it?
Yeah.
Yeah. So that seems quite odd.
Yeah, he used antibiotics before, right?
And did it work?
Well, so he's sequencing before and after, and continuing to, and we're waiting for results. The entire DIY bio community is waiting for his results.
When did he do it?
We're waiting, Josiah. Like, I don't know, a few months ago. I think it was featured on Wired or one of the big magazines. Sorry.
No, that's all right. So, yeah. So we did have people coming from in the States to have it. But in the scientists I've been working with, in 90% of cases, after a maximum of 2 treatments, patients are cured of Clostridium difficile infection by this method. I met a scientist recently who at Wellcome Sanger Institute who is using culture methods to find lots of new strains of gut bacteria and sequencing that, and he hopes to get it down to a kind of hill with maybe 11 bacteria that you can take. At the moment, it's kind of an unpleasant treatment. So I should explain the artwork. So the inside the glass colon there is a sterilised human fecal transplant that I made in the lab. It's not my gut bacteria, it's the scientist I worked with. It's her gut bacteria. It's sterilised because it has to be sterilised, otherwise it will explode in the gallery, and you don't want that. And that would be quite dangerous as well. It's quite nice because I visited it again about a year later, and it's because of the way it's kind of moving around inside, it's kind of gone a kind of reddy colour now, which is quite nice. It's sort of changing colour.
And I just got to say it's one of your shittiest art.
Thank you.
Yeah.
You wait till you see the new
Yeah.
one I'm working on. So the case is carved with the spores. So Clostridium difficile, it forms spores. It's a sporing bacterium, and they think that one of the main reasons for reinfections is that the spores are held in biofilms in the lining of the gut, and so when you move around and stuff like that, that breaks down, and the spores are re-released into the gut. So the scientist I was talking about who wants to make the pill with the 11 bacteria, 11 he thinks at the moment, he'd be working with spores with that, and he's got this idea of spreading health through kind of beneficial spores. But that's not the people in Leeds that I worked with on this. This is the Healthcare Associated Infection Control Team at Leeds University. They have a huge gut model, and they kind of kidnapped me. I think it's fair to say. You're recording this, aren't you?
You're live on Facebook.
Okay. They might actually be watching. So they kind of kidnapped me, I think it's fair to say, and they invited me up to give a talk, and after I gave my talk they said, we'd like you to come and see our gut model. And I thought, oh my god, this is going to be really boring. It's going to be one of those computer models. I'm going to be sitting there watching them change parameters for like 2 hours, where they go, if you change this, it does this. Because I've had that experience before. But it wasn't. It was a whole room full of beautiful bubbling jars of pink liquid and things and stuff. And they started off with a healthy gut sample, and then after it's all kind of bubbling along and growing nicely, they give it a C. diff infection, and then it still carries on perfectly happily. And then a bit later, once it's all growing together as a big community, they give it antibiotics, and then they observe how it changes over time, and they study that. And they've got one jar where they're studying, it's got glass rods in it, and they're studying how the biofilms form, so they can look at those and how the spores form in it. So this sort of intestine-y bit here is made from cloth that was impregnated with the different gut bacteria, so it's things like Enterococcus, E. coli, C. diff, on different chromogenic agars, and inside it is one of the tubes that they use in their gut model, which is also what they use for their human fecal transplants. But it's kind of an unpleasant procedure. You've got 2 options really. You can either go down or go up. They prefer to go up because there's less risk. Not in Leeds, but a patient did get I think a Klebsiella pneumonia from a one going down that went wrong, so a really nasty kind of gut bacteria related pneumonia in their lungs, which is not a good thing, so they prefer to go the other way. Of course there's problems, so it's not suitable for everyone as well. So a lot of the patients who need a fecal transplant for C. diff infection are elderly. Some of them might have Alzheimer's disease and things like that, so it's not possible to really perform such a procedure. It's quite unpleasant in a way. It's not that bad, but if they don't fully understand what's going on, you can't just do something like that. So it'd be really nice if it's possible to come up with a kind of different method of doing it to treat people better. Yeah, so that was that. The gut microbiome is kind of fascinating, and that's what I'm kind of working on here. This is a piece I made with the UCL iGEM team, and it's a probiotic E. coli. It's a lipstick here with a probiotic E. coli that has a plasmid on it that codes for chloramphenicol resistance, and on that plasmid is something that makes it produce serotonin in the gut.
Wow.
So it's a probiotic E. coli.
They have the whole pathway of serotonin.
Yeah. So
okay.
Well, as I understand it, yes, it will produce
very
serotonin in the gut. So the drawbacks with this, obviously, are that one, I'm not allowed to take this artwork out of the lab because of all the very strict GM laws in the UK at the moment, and two, it's on a plasmid that codes for chloramphenicol, which is quite a nasty antibiotic. So if you're not taking the antibiotic, then it won't work. But that's sort of like intentional because it's a kill switch, really. So it's this way of like, it won't grow in you if you're not taking chloramphenicol. So it needs that. It's sort of like in Jurassic Park. It needs to be in the presence of chloramphenicol for it to keep the plasmid. So the lipstick's got it's freeze dried. I freeze dried the bacteria because I worked with a scientist many years ago who was obsessed with probiotics, and she said that yogurt drinks are rubbish. And she thought we should all have salt and pepper pots on the table with different gut bacteria and with different probiotic bacteria on it. And I think that research is still going forward, but the freeze drying was definitely the best way to do it. So it's the lipstick contains freeze dried bacteria. That's a food supplement you can just sprinkle on. And up there, these are jellies, but they do contain chloramphenicol, so you could take it if you took one of those, and you've got the whole thing. Obviously, I don't recommend anybody does at this stage because one, chloramphenicol has really nasty side effects, and two, they never tested what quantities it's going to produce the bacteria, the serotonin in your gut, and so we don't know. Like, if you had a huge overproduction of serotonin, you might not stop. It might be like a C. diff infection or something where it starts to proliferate. So it's a kind of proof of concept that we collaborated on there. Whole genome sequencing has kind of revolutionised microbiology hugely, and this dress is called the sequence dress, and I made it in collaboration with Alex May at the back, and we used his software for it as well. And I sequenced from end to end the whole genome of the Staphylococcus aureus that lives on my body. The scientists I'm working with are kind of obsessed with Staph aureus, and it's resistant to penicillin, so but that's kind of normal because 80% of Staphylococcus aureus is resistant to penicillin. And the dress is like the quilt impregnated with the Staphylococcus aureus, the cloth impregnated with the Staphylococcus aureus from my body, with MRSA and with something called VRSA, VRSA, which is the vancomycin resistant Staph aureus. So that one's resistant to your sort of last ditch antibiotic. And the projection behind is the whole genome of the Staph from my body. And on the dress, this projection, this kind of blobby projection that's video mapped onto the dress, that's the light output of the Illumina sequencer that we use. We used a MySeq sequencer, and it produces, because the DNA is kind of attached to fluorophores which fluoresce in different colours depending on, say, A, T, G or C, and you get photographs of this. So you can actually thread back together these photographs. And so we made a film of that, so you can actually kind of experience the whole genome in light. And it was quite strange to me to realise that something so complicated as a whole genome sequencer was a light optical device, really. And then we made a virtual reality version of it, which has been quite popular. So you can can actually kind of travel through the light output of the whole genome sequencer as it's sequencing my Staph aureus and inside the DNA. It's kind of got a narrative, so it's quite an educational piece, that one, but it goes alongside other pieces, and it's very useful for the scientists, because we use it in museums and art museums like the Victoria and Albert Museum in London. This piece was made in collaboration with the Liu Lab for Synthetic Evolution at University of California, Irvine, and it's based on an engineered antibody which binds quite well to HIV. So they're working with synthetic biology, and they're working with kind of non-canonical amino acids. So this antibody that they were working on contained 21 amino acids, and life normally contains 20 amino acids. So each of the beads in the necklace contains the actual amino acid in the sequence in a crystallized form, so they're polymer clay beads. And so it's the entire sequence of the amino acid sequence of the antibody. And the loops there are the actual folds in the protein. So you could actually rebuild the synthetic antibody from the necklace. And the background of it is dyed with Kumasi blue, which was originally a wool dye, but is now used in things like synthetic biology to look at DNA. And there's some other details. And I also put the engineered antibody into an E. coli. So this is like the genome of bacteria is circular. So we kind of thought of that a bit like a necklace. So the other piece, also some people might say I kind of hate like the curators of galleries in a way, because I don't, but it's like, it seems like I do, because that piece was called engineered antibody, the other one. This one's called necklace. So it's really hard for them to know where to put the labels. And I always get it wrong. I have to take it down. And so I'd like to think of the ring genome of the bacteria like a necklace, and it's like we pulled apart some of the beads and we forced in the actual engineered antibody, so it's in the genome of the E. coli, and then it's got one random amino acid change, which means it either grows blue or white in the presence of X-gal. So if you've got one jar of bacteria, one jar of bacteria, and it's exactly the same bacteria apart from one amino acid change, which means that they're blue or white, which I thought was kind of amazing, really. Just one, such a tiny change. Sort of coming towards the end, a couple of final projects. I worked with, and I'm still working with, Professor Maggie Smith at the University of York in the UK, who's working with Streptomyces bacteria and using something called phage integrases, which are the bits of virus DNA that kind of bind into bacteria. Sometimes they're usually, so I should say what phages are really. So phages are viruses that infect bacteria, and they look like little rocket ships, if you don't know, and they land on the outside of the bacteria, and they kind of put a corkscrew into the bacteria. Normally they inject their DNA and hijack the cells' mechanisms to produce lots of copies. Sorry?
The thing that encodes the DNA
Polymerase. DNA polymerase.
Then, but some of them don't do that. Some of them just integrate into the DNA of the bacteria and just hang about there, and then only do that when the cell seems to be in trouble. And the bacteria seem to don't mind them being there because it sort of helps them recognize other attacks. So she found that the bits where the phage integrates with the bacterial genome, you could use that kind of like Lego to slot other bits in. So it's, I mean, she discovered this about 20 years ago, but for editing antibiotic pathways in Streptomyces, it's more effective than something like CRISPR by a long way. So she's still working on developing this, and it's a textile piece which is impregnated with Streptomyces colour, which we edited using these phage integrases. So it only produces a blue antibiotic and not a red antibiotic. Normally it will produce a mixture of blue and red, but this one won't produce the red because that's been edited out. So potentially with this work that she's doing, you could you could say, well, I want to edit the antibiotic pathway of this streptomyces, but I want it to do this and this that those other bacteria do. And you'd be able to kind of plug that in to the antibiotic pathway of the streptomyces to have a kind of super antibiotic producing bacteria, which would be good, I think. But antibiotic resistance is still a huge problem, and this is kind of like close to where I'm at the moment. So this is something I'm working on at the moment. These are kind of like votive offerings, like you would see in like a Greek church. Where have you ever seen these things? Do you know what I mean?
Green church?
Greek church.
Greek church.
Like Greek Orthodox churches or Catholic churches sometimes. So when people kind of, if you had like a bad leg, you'd get a kind of like a piece of silver that had a picture of a leg on it, and you'd go to the church and you'd hang it up.
Yeah.
And that either means that you want your leg healed, or you're kind of giving thanks for your leg being healed, or you see them with eyes on, or you see them with soldiers on and different things. So I've been, in a kind of non-religious secular way, we're really interested in like the issue of antibiotic resistance and kind of collecting people's stories about that, because the Wellcome Trust in the UK produced a report about antibiotic resistance, and they said that statistics make no difference to people's opinions on antibiotic resistance, and it's stories that they need. So one story is much more convincing. I suppose this is sort of kind of obviously kind of played out in politics these days, isn't it? A story versus statistics. Yeah, people don't care. So we've been kind of collecting people's stories about using antibiotics or about bacteria and about different areas of research if they're scientists and things. And they make one of these votive offerings out of a kind of aluminium, and then they're hung on these ribbons, and all the ribbons are decorated with or impregnated with different bacteria. So this is that streptomyces there. This one you can't really see very clearly in the picture, but this is diverse colonies of gut bacteria on chromogenic agar, pinks and blues. And this one up here, the orange, that's Prontosil, which is the kind of first antibiotic, like chemically synthesized antibiotic. It predates penicillin, and it was produced by Gerhard Domack based on Paul Ehrlich's ideas of the magic bullet. So they're made of lots of different antibiotics, so all the ribbons are different. So this is a new participatory work that I'm doing that's growing over the next like 8 months, and it's in Oxford Museum of History of Science, where they've got a show called Back from the Dead, Demystifying Antibiotics, which is commemorating, it's the 75th year, the 75th anniversary of the first use of penicillin in a human patient. Now, I'm sure you've all heard of Alexander Fleming, but have you heard of Florian Chain? Have you ever heard of them? So they won the Nobel Prize with Fleming. A lot of people don't realise that Fleming actually abandoned his work on penicillin after kind of discovering it. He spotted it, he was looking for antibiotics. There's sort of a bit of a myth that he kind of randomly went in the lab and noticed it. He was looking, but he said it was way too hard to work with, and he abandoned his research in about 1928. But Florian Chain, who were two professors at Oxford, worked on it, and they actually got it working, and then they collaborated with scientists in America to produce it in much larger quantities. So it was first trialled in the UK by Florian Chain in Oxford, and they're very little known, because I think they didn't really care about the publicity in the same way, but it's really interesting. So they all won the Nobel Prize, and then Dorothy Hodgkins was also hugely important, and she won a Nobel Prize later on as well, but she was the X-ray crystallographer that kind of discovered how it looks, the molecular structure of it as well. And so all these people are kind of unseen. And so this artwork is in an exhibition recounting this whole history. And so people come to this show, and they've actually got, so with penicillin, apparently it was like having a boiling water put into your body through a very thick needle in the early days when patients were treated it. And with the first patient who had a staphylococcus and streptococcus infection, they could only keep him alive for 3 weeks because they couldn't produce it in large enough quantities. And they were, because like with vitamins, you pee most of it out. They were keeping his urine, and they were taking it back to the lab, and they were re-extracting the penicillin and giving it back to him. And the patients who received the penicillin that had been through somebody's body said it was much more pleasant than the fresh stuff, so to speak, as well. So there's so many amazing stories. And they've actually got some vials of the patient's urine in the exhibition. But what's really important about fluorine chain, it was during the Second World War. This will be of interest too. It was during the Second World War they were working, and they couldn't get any lab equipment. So actually their culture vessels, and this is at Oxford University, one of their culture vessels is actually, and they've got it in the show, 3 biscuit tins welded together with tin cans stuck on the outside. Other things that they would culture penicillin in, they would actually use bedpans with lids to culture the penicillin in, so in the early stages. And they were so worried that the lab would be bombed during the war that they all kept stocks of penicillin mould in their pockets, in the lining of their pockets, so they would always have it available if they needed it. So it's a fascinating story with them. So this is a growing piece, and it will grow over the next 8 months, and then I'll have a solo show in the gallery, and most of these works that I've talked about will be there. And there's some other close-ups. Now, if people want to, I've actually got some metal with me, and you could make one, and we could add it to the installation in Oxford, so it could be part of that. And then just one final piece that I just finished last week. I think it's arguable, because I've never looked into whether any others exist, but I think it's the first lab-grown tooth made of bacteria. So it was a commission for the Science Gallery London, and in collaboration with Rachel Salmons and Melissa Grant again, and it's a Serratia N14, which is an extremophile bacteria that lives at the sites of highly polluted mining areas, and it has this way of kind of pushing these toxic substances out of itself. So what we've made it do is produce hydroxyapatite, which is basically tooth enamel. So we grew it in a bioreactor on a piece of sponge that was cut in the shape of a tooth, and the bacteria grow its biofilms all over it, and then we treat it with the minerals that it needs to kind of push back out, and then we fire it in a kiln at 1,000 degrees to get rid of all the foam, and then you're just left with the hydroxyapatite produced by the bacteria. And then we did a participatory thing alongside it where members of the public could paint their own porcelain teeth, but all the glazes on these are made with the bacteria that cause tooth decay and gum disease, so Porphyromonas gingivalis and Streptococcus. And that's kind of what it looked like in the end. It's on show now, and so is the other piece. And the secret stress is also on show. They're all on show in the UK at the moment. They're unfortunately a bit far from here, but if you haven't been on your way there, then that's it. Thank you.
Anna, Dumitriu, thank you.
So if there are
You guys have any questions?
I'm happy to answer.
Small question.
Yeah.
What kind of biofilm was on the?
It's a biofilm from Serratia N14.
You know what material it is?
In
Like cellulosic or something else?
I don't, but I could find out. It's a bacterial biofilm. But actually, I've got some scanning electron microscope images. So, yeah.
Isn't all this bacteria dangerous at a certain point?
Yeah, they're dangerous at a certain point. They're not dangerous when I exhibit them to the public. They would be dangerous if I was handing them.
So what's you kill them off or something?
Yeah, yeah.
Because I'm thinking, these people are painting with live bacteria.
Oh no, so they're not. So that was the pigments were made with the bacteria, but it was again autoclaved. So the colour remains, but the bacteria are dead.
And you didn't have any problems putting DNA on stuff and presenting it? So
No.
DNA is inert
No, so I
to,
checked.
to science.
So it's a validated kill protocol by Public Health England, the TB DNA extraction. So what they needed to do was prove that you could work outside of a Category 3 lab with TB DNA.
Right.
So they did that.
And that's fine.
The scientists I worked with did that. They validated the kill protocol. I did the same method with them.
And the kill protocols, it means that
It
TB
means
outside
it's
of the cell, it will not do
means,
anything.
It means they've tried it thousands of times and you can't possibly grow
Yeah.
any live bacteria
Yeah.
from the DNA. There's no trace of the bacteria left in the DNA.
So you're dealing with billions and billions of bacteria?
Yeah, yes,
Yeah.
all the time.
No, I think what he means is that you're dealing with more bacteria than the usual person. When you're doing a big art piece, when you're growing like
Oh, yeah,
50
I mean-
liters of bacteria,
yeah.
no one really gets to do that.
Yeah.
You know, to be
Yeah.
around that number of bacteria.
Yeah. Well, I've done pieces on quorum sensing as well, so I'm well aware of the issues related to that too. Yeah, so I work the sort of the health and safety in the work. If you're sort of talking about that, I actually work with the leading scientists in the UK on my kind of kill protocols and sterilisation, all my processes. After you turn that off, I can name some names.
Are there other artists doing similar stuff?
There are. There's Dumitriu here who works with bacteria as well. I mean, that's similar. It's not similar, but it's working with bacteria.
And
So
we also have Oli in front of her.
Yeah.
Oli, say hi. We did a great project from Betzalel about growing bacteria in print, and so printing the Hebrew letters in bacteria. Yeah.
I think I've seen that.
Yeah.
Live.
3D printing with bacteria.
They already 3D print the bacteria, so you know. I think so.
I think it's with the agar. It's still quite fiddly. There's biohackers say they're doing it, but like Varg Society in Amsterdam did an event on that, and they said
there is a bioprinter, but
yeah. I think it's harder than
syringes and printing is hard.
Yeah.
You can do it, but it constantly needs to be calibrated.
So I think all they were doing was 3D printing the agar, which is still quite fiddly, and then growing bacteria on structures.
and it's 2D, I think. You know, it's just like
As a 2D.
it's just a one layer of bacteria that you can choose where to put what cell. But you know, you talked about spores, right? And your friend with 11 strains of bacteria to fight C. diff. So there's one company called Ceres Health. You familiar with them? So from Boston. And they were the leading company in doing the spore peel to fight off C. diff, and they failed recently their clinical trials. And I think it's mainly because of the complexity of the issue they're tackling, and the fact that current regulations are not compatible with dealing with such questions that involve such a complexity. So different people will react differently. So when you're trying to do a clinical trial, you have to choose the different people, but we don't know how to choose the right group of people to make it work. So it's not working. But yeah, there is great hope in the microbiome field. Anyone here in biology in working on microbiome stuff? Do you have anything to say?
I guess I work more on the oral microbiome, so a lot of people focus on the gut just because of your numbers, but I was very interested in the work you did showed in the end.
Yeah, so you're not leaving it out. No, it is important.
Actually, I was wondering, how do you see the interface like with the real scientific, those who are not in the art, but rather conferences and scientific papers? You get all your data from talking to scientists, but what about getting information from all the other platforms in which this information is shared, like conferences and
So yeah, I mean, I go to things like that. I mean, every year I go to the Modernising Medical Microbiology conference and absorb that. And it took me I think it took me 3 years of talking to the scientists and going to all these conferences on sequencing to even dare to tackle whole genome sequencing. I was still at the stage of like, I don't really understand this. The only viable method I think of for learning this is to actually learn, like get all the scientists in the whole pipeline to teach me what they do and me do it hands-on, because that's the only way I can learn enough to be able to tell somebody else about it and answer their questions. So that was sort of the first thing I did. So I do go to conferences and different events and
What are the
talks
responses you get?
online.
People are
They-
surprised that people do art with these. With
Well, I
their
don't know.
stuff?
So usually I get some invitations. So I mean it varies. But I don't usually get a bad reaction. I haven't. I haven't for a long time anyway.
I hope it keeps up.
Yeah.
So as a kid, I remember I've been to a class where we took a petri dish and placed our hands on it. Then we washed our hands with soap, put it again, put it in the incubator for a couple of days, and saw the marvelous colonies that grew out of it. Do you think this sort of experiment would pass the criteria that you have in your art installations?
So did you do that in what setting were you in doing that?
Home, a classroom.
Oh, in a classroom in a school?
Yeah.
So, I mean, usually they kind of doing the strict methods to dispose of things, and they seal the plates afterwards.
No,
and then they
it
don't let
was
people
school.
open the plates afterwards. I mean, you sort of can. It depends what agar you grow it on and what temperature you grow it at. So, with the things that are cultured through the DIY method, we culture it under 25 degrees to slightly reduce the risk of growing human pathogens. And the agar that I use is a non-selective agar that Simon Park created, that's actually made from supermarket products. It's made from, it's the agar Marmite, this UK delicacy.
Which is yeast extract.
Vegemite, skim milk powder and honey. So it's actually edible. So legally it's food. So you can just throw it in the bin. And we're not trying to culture pathogens in that way, and it probably wouldn't be a very effective method of doing it. So the scientists that I work with have said that this is kind of okay to do. Alex and I made a film of our skin microbiome growing, but we were very, very careful with it. So I always treat it as if I'm doing do-it-yourself biology kind of at home or in my studio, then I always treat it as if it's the most deadly thing. So I have handled pretty much the most deadly bacteria, so I've worked with them, and I would treat it in the same way, pretty much. I don't have a category 3 lab at home. I treat it as if it's really nasty and seal it up and throw it away. But it's no worse than a bit of raw chicken. I mean, in terms of risk.
Mm-hmm.
And I'm not even talking about a bit of gone off raw chicken, I'm talking about a bit of fresh raw chicken. It's no worse. So, but of course, you've got that quantity thing that you raised the issue of.
Yeah.
When you can see them, you've grown loads. So it's far more probably than you need to kind of create a bad infection. So you have to be extra careful. I do this workshop. Nuri had me do it at Jen's Space in New York, where I basically say that I'm trying to put the fear of God of people, I send people away absolutely terrified of what they're trying to do, but with hopefully all the knowledge of what you need to not kill yourself. So I've even done a workshop where I called it like how to do it yourself microbiology without killing yourself, or others in brackets, which I think is very important, isn't it? So that shouldn't be
The artwork that you showed that it produces serotonin.
Oh yeah.
You said that you could not show that, or
so in UK, there's you need a license to handle GMOs to display it. So the gallery can, like, if you had an institution that was willing to extend
their license to handle GMOs to a gallery space, then you could show it. That's a little bit difficult. Howard Boland did it in the UK. He had an exhibition at the Royal Institution in London. So he showed some synthetic biology artworks.
Even that's crazy. Even though you just said that you showed like tuberculosis, which
I
you
sterilized it. It's not live.
So wait, okay,
No,
so you could show it.
Extracted
Okay.
DNA of the TB, so it's not the TB's not. In that case it's the extracted DNA of
Okay.
TB. In some other pieces it's sterilized bacteria that I actually exhibit live. So what I would like to do, and I've had this ongoing dream of kind of building a space where you could show stuff like that in an art gallery live, and maybe even have members of the public come in and work with me to make things with pathogenic bacteria, work with a microbiologist and myself, to safely do it, because they could do it. You know, a lot of you all know that you could quite simply do a thing if you were being looked after by some people helping you. And by doing it in an art gallery, I think you'd have a different experience of it, because I'd want people to come to it so they weren't just thinking about the science, but they were also thinking about the aesthetic of it, like how does it smell, how does it feel to be a scientist?
So why not work with the good bacteria? Why work with the pathogenic?
So yes, you could do that, but over the years I've kind of built up this, I guess, level of trust working with scientists, and there aren't really that many artists working with clinical microbiology with pathogens, and so in terms of engaging the public in the issues around it, that kind of would be left, and I think that's really important. I think being able to talk to people about the pathogenic bacteria that harm them is a really important area for them to discuss, because people, like older people in England, are scared to go to hospital or have an operation that they need because they don't have anyone to ask about MRSA, MRSA. And so we did workshops where I had the scientists who are the experts in MRSA, who are also doctors, and we did MRSA quilt making. And then we had participants of all ages. We did it at the Victoria and Albert Museum, and they could come and they could make a quilt square, and then we took it back to the lab and inoculated it with MRSA bacteria, and they could see the results of their own experiments of how different antibiotics would work on it. And then that was sterilised and made into another artwork. But what happened, and it's the same with making these kind of votives, is conversations happen, and people, it breaks down the hierarchy between the scientists and the members of the public, and people feel happy to chat and ask kind of silly questions that they're frightened to ask in other settings. So I think it's really important to work with these medically important bacteria that are a problem for us, to kind of get those issues out. And antibiotic resistance is a massive issue.
Yeah, thank you. That was very well said. Yeah, I think that in general, in diagnostics, for example, what we try to do is take something that is invisible to the human eye and make it visible in some way. So make it color, give me a signal. And then the human can sense it and understand it. So when we're talking to people, like you say, older people, they don't know how to understand this micro scale. So using art for that is beautiful.
Doctors in hospitals, they're telling patients, you better off leave the hospital because you might get a disease here.
Mm-hmm.
It's all contaminated.
Well, they don't even know how Staph aureus, the vectors of it, how it spreads, so how MRSA spreads in the hospital. That's not even clear. There's been big studies. There's only been like one incidence where a healthcare worker passed it on to a patient. And where else is it coming? How else is it coming? They still don't know, which you'd think with all the tools that we have. Now, people I know are working on various things around that, but the fact that we still don't know where it's coming from is kind of fascinating.
There's been an interesting study of patients. They came into the hospital and stayed in a bed that the previous patient had they used antibiotics on him. So if he developed a drug-resistant bacteria, then the patient who came in, he might get it.
Did they show you that was
Yeah,
happening?
yeah.
They showed that a patient actually acquired it from the
The
bed.
patient acquired it from the bed of the previous patient.
That's quite bad. So they in Brighton, which is a big centre for Staph aureus research, they did a big study on the intensive care unit, and they literally swabbed everything, and they swabbed all the staff and all the nurses and all the patients, and they couldn't identify a clear vector for it. So either it's coming in that case, either it was coming in on visitors or other things that that were coming in and out that they couldn't control. It's not known, but it left more questions than answers. And some people in Cambridge showed it coming from one healthcare worker. They had a healthcare worker who was infected with MRSA, but there was no transmission event, so nobody caught it in the Brighton study. So then the healthcare worker was treated, but there was no transmission event. They didn't give it to a patient. So it's still not clear. When we stop recording, I'll say.
Okay. Wow. Okay. Really? Yeah. Okay. I'm comfortable with this. Okay. So I have two things to say. One, bacteria, they're not bad. Okay. They're okay. They're
mostly harmless too.
Yeah, we started everything alive kind of started from single-celled organisms. Okay, and the term pathogen, so a bad bacteria, is a contextual term. It means that a bacteria is bad only in a specific environment. A bacteria that is bad for me can be good for you to some extent, right? And you talked a little bit about elements, genetic elements moving. So an antibiotic-resistant gene is an element, and it can move from different organisms. So it's called horizontal gene transfer, right? And we know that small fragments of DNA called plasmids can move it, and viruses can move different parts of DNA between microbial cells, and even randomly it can happen. Or through a process called conjugation, which is when two bacteria come and they kind of kiss and they move DNA to each other. And in this process that is kind of, we talk about it here like it's bad, it's moving the resistant gene. It's also a very strong force in evolution in general from the beginning of time. So all the time, and when we're looking at these things, these small things, we have to remember that they're not bad, they're a basic part of life, and we're just lucky enough to begin to understand them and study them and work with them. And when we use art to articulate this, I think it's a great way to get the public engaged. Like you said, really exciting stuff. Okay?
Thank you.
Thank you for coming.